is Caroline. She needs no introduction, of course. Many of you know her. She's been at MIT since 2015. She's part of IDSS and the LIDS and the Center of Statistics, and she's interested in statistics, machine learning, computational biology. And many of you know that she's working on graphical models, causal inference, algebraic statistics, and applications to genomics, for instance. And today she's going to do the impossible, and she'll talk about autoencoders and COVID. And we're looking forward to that talk. And importantly, she is now in Zurich, where it's 5 p.m. Is that true? And so we're making sure that she is self-isolating on this Friday evening.
Thank you very much, Sasha. Yeah, so let me just get started with kind of the motivation for this talk and describing the kinds of problems that we've been spending quite a bit of time thinking about, or the kinds of applications that we've been thinking about, and that really led to all of the theoretical work that we've been doing. So in general, these problems are problems of multi-domain data integration and translation. So while we come from, the problems we look at are mainly biologically motivated, these same problems appear in many different domains. So think of, for example, you have audio data and you have video data, and you would really like to integrate it for spatial correspondence, right? For example, for getting better object detection. Or you have different kinds of data modalities like video, radar, lidar, and say you have corrupted data in some of the modalities, and now you would like to use the others in order to predict the corrupted modalities to compensate for it. And the same questions arise in biology. Those are the kinds of questions that we study. The problem in single-cell biology is that usually getting the data means destroying the cell. So that means you cannot look at different modalities of data in the same cell. Okay, so either I can decide to take an image of a cell, or I can decide to look at its expression, but I cannot do both in the same cell because I have to destroy the cell to get either one of the two. So that means, in order to study like biologically what happens, or how are these two things, modalities related, like expression and spatial things inside the cell, you really need to be able to translate from one to the other. And the other thing where similar translation and integration problems arise is, nowadays there's a whole lot of interventional data available, right? Think of online education, or, just A/B testing, like for example, in personalized ads, or in manufacturing, etc. But also, of course, in the biological domain where, you have all these different drugs, and right now that's super important, right? We do a whole lot of drug screening for finding a drug that maybe can be repurposed for SARS-CoV-2. And there you also have a huge data integration and translation problem. In particular, many of these drugs have been applied to particular cell types, but now we care about this one particular cell type in the lung, right? And we would want to figure out, and in particular for this one particular cell type in the lung that is diseased in this particular way, and we would like to figure out which drug will actually be able to revert it back to the normal state, even though we haven't measured that, right? Or also you want to be able to do this at a person-specific level, and maybe you cannot measure this on all people, but you would like to be able to translate the effect of a drug from one person to another. Similarly, you may want to translate between different interventions. So for example, in genomics, we can now actually go in and intervene at any one of the genes, but humans have 20,000 genes. So obviously we'll not be able to do all interventions or knockouts in this case. So you'd really like to, if you have data from 1 or 2 or maybe even 10 or 100 interventions, you would like to be able to predict what happens if I intervene on a gene that I have never intervened on, just based on all of the data that I have. Okay, so these are all multi-domain data integration and translation questions, and I will now be quite specific in the 4 questions that I want to look at and will be looking at. And these are all the kinds of questions that I already discussed on the previous slide. So here they're written out in terms of biology questions, but obviously they can be just translated to other kinds of domains. So the first one here that I have is a translation question between different data modalities. So say I have a whole population of cells and I cannot measure both modalities that I care about at the same time. So I take some cells out and I send them to sequencing. So that's here the data I get. So these are my samples, and every one of these rows is a vector, is an observation vector. It's the expression of all of the genes in this cell. And I take other cells from this population and I send them to imaging, and these are the kinds of images you get out. And now what you would really like to be able to do is, have this map, right, that goes over here from expression, RNA-seq to images, where I can take a particular cell and its expression profile, and this will predict to me how this image, the corresponding image for this particular cell, would have looked like were I able to also image it. Okay, or the other way around. So that's one question. A quite similar question is this one over here, where I have different time points, right? So because getting data is destructive in all these biological modalities, it means you'll never get the standard time series data, right? I cannot observe a cell over time throughout the process that I might care about, like, for example, cancer development or something like that, right? So I can have a population of cells which goes through a process, and I can take out some at time point 1, but then I'm destroying them when I'm getting this kind of data. Then I can take out some others at time point 2, I'm again destroying them. But again, I would like to be able to ask the question, I'm taking out a particular cell. How would this cell have looked like at time point 2? Were I also able to get it? Or of course, the other way around, right? So often you want to be able to look backwards in time. So this is known as lineage tracing, right? I would like to be able to predict or generate a particular image for this cell, how it would look like forward or backwards in time. So that's all at the observational level. Then similar questions, and this is the question that is relevant for SARS-CoV-2 that I talked about, is at the interventional level. Here I'm doing it in a different context, maybe not SARS-CoV-2, but the kind of thing that pharma industry would really like to be able to do is the following, right? They do all these experiments, they have all these different pharmacological inhibitors and all these different drugs, and they do these experiments on mice, right? And obviously you always have a whole lot of control samples that's just without any drug, and then you have all these different drugs that were tried out in different disease contexts, and now you want to translate it to humans, right? And so in humans you of course have all this control data, and now what you would like to be able to do is predict what happens, right, when you apply any of these drugs in the human context. So that's, of course, an interventional question, but again, it actually looks quite similar to these questions up here, right? Because somehow here you always have these questions about 2 populations, 2 distributions. Like here I have a distribution and a distribution. I know they kind of have to match because they're from the same population of cells. It's just 2 different modalities, right? So I kind of have this transport problem between different distributions. Same here, right? I know these 2 time points. Well, I want to somehow, a cell shouldn't move too much, right, from one time point to another. So it's again a question of matching distributions. And then learning this function here. Similar here, right? You're in the control setting. You want to learn this function here, and then hopefully the same function or a similar function will allow me to go also when I am applying a drug over to here. But here is a very different kind of problem, or at least we haven't seen how to turn this problem here into a problem that looks more similar to these ones up here. It's the following interventional problem, and I would love to hear if someone would know how to do this. But so it's the following interventional problem that I talked about knockout interventions. So this is what I'm representing here. So a cell, human cell, has 20,000 genes. So you can go out and just set any one of the genes that you like to 0. Okay, you just knock that gene out, and you get to see how does the expression profile look like of the cell after you do that. Okay, so control here just means observational. I didn't intervene at all. And then, you know, you have all these different interventions. I can go knock out gene 1, gene 2, gene 3, combination of genes. But obviously, since I have 20,000, right, I will not be able to do all of them. And so what I would really, really like to be able to do is go over here where I have, you know, nothing kind of to match, right? And I would like to be able to say, well, you know, here is a knockout combination or a particular knockout of a single gene that I have not seen. Well, can I predict it, right? I have all this data over here. Can I go from all these different kinds of perturbations that I have seen to a completely different perturbation where I haven't seen anything? So this seems, at least, I mean, to us, we have been using very, very different methods to actually look at this problem as compared to these three problems here. For now, it seems like here we really have to learn, and here we're taking a graphical models approach. We kind of have to learn from the interventions that we have here, the underlying, in this case, causal graph, the full causal graph, in fact, if you want to be able to predict any intervention. And once you have, and you see here, I drew a little network, right? Once I know this kind of network that I can learn from all of this data over here, well, then, you know, if I know where all these genes are that I'm going to intervene on, well, then from there, I can actually go down and predict what happens when I knock out these other genes. Okay, so to us, this kind of problem here was a very different problem than these, although we would love to be able to solve it a bit differently because this is hugely sample and computation. You know, it needs just a huge amount of, not very sample and computationally efficient, let's say it that way. And so as compared to, you know, the other kinds of approaches that I have here. Now, because every talk I've given at MIT has been on graphical models, what I thought is I will not talk at all about anything down here, about this graphical models framework. But what I want to talk about instead is about these three other problems that we've looked at. In particular, for this autoencoders setting, I will in particular talk about this one paper that just appeared, oops, sorry, I'm going forward, and that actually was just accepted this week to PNAS, which is with Adit Misha Belkin on overparameterized autoencoders. Just because the autoencoders are in fact the backbone of all of these methods that we're using in order to match these different kinds of distributions. Okay, so what I want to do is first tell you a bit about, you know, why do these autoencoders work, so that we understand that much better, and then that can maybe inform also how we actually want to do these kinds of applications afterwards. Okay, so that's the kind of outline for this talk. So I'll first do the theoretical parts on autoencoders and then get to these three different applications. And in particular, the theory will inform this last application here, which is the SARS-CoV-2 application. Okay, so what is it? So autoencoders, I'm sure everyone is familiar with. So these are very nice neural networks. There may be more special neural networks in the sense they're not the standard classification neural networks. So what they do is they learn a function from some input space. Say, for me, this is going to be RK or RK0 in this case, to some output space, which is again RK0. Okay. So for example, here, if you put in an image, the same sized image will be coming out. And you train these autoencoders so to minimize a reconstruction loss. Okay, so you have some training examples, in this case, say n training examples. And what I want to do is to minimize psi of xi, so what comes out over here minus what I'm putting in this case, into a norm. Now, usually you go through a lower dimensional latent space in the middle because you don't want this function to just learn the identity map, because that's maybe not as useful. So you're going through this latent space to hopefully get some nice representation of your data in this lower dimensional space. Now, what I want to talk about is actually the setting where maybe this latent space is not lower dimensional. So maybe actually this latent space is even the same dimension of the input space, or even higher dimensional, so that you could learn the identity function. And I want to show that that's actually maybe useful. Okay, in particular, that you don't need this regularization here going through this lower dimensional space, but that even going through a higher dimensional space, just training the autoencoder itself already adds regularization and might actually lead to something very, very useful in terms of a representation that you get of your data. Okay, so that's kind of the whole idea of this project, is to look at what happens in this overparameterized setting when I might not constrain the network architecture actually to go through something lower dimensional. Okay, so let's look at the problem itself in a very simplified setting. Okay, so this is the problem that we're looking at, right? So from the previous slide, so I have my n training examples, say they are in RK. And let me assume that n, so the number of samples, is smaller than the dimension in which they live in. Okay, so in this case, I am overparameterized. And now, in fact, in particular, I can get this loss function down to 0, right? And so usually autoencoders are trained using some stochastic gradient descent here. Of course, when we do a theoretical analysis, we're usually looking at gradient descent first. So I look at gradient descent methods, and they're usually initialized close to 0. Okay, and so they're initialized to minimize this loss here. And as I said, in the overparameterized setting, this loss can be 0, right? So let's look at the very simple setting first, just to get some intuition, when in fact this function psi here is just linear. Okay, so think of psi just as a matrix multiplication, right? Say this is just some matrix A, so I'm looking at A times xi minus xi, 2 norm, and the sum over all of these. Okay, so I can get this down to 0, right? Because I'm in this overparameterized setting. Now, of course, we know that there's many, many solutions, right, of matrices A, for which A times xi, or let's do it even for just one sample, right, for which A times x minus x is equal to 0. Right? In particular, for example, the identity matrix, which satisfy this, right? The identity matrix would just have xi minus xi. Of course, every one of them is equal to 0. But there are many other solutions, right? So for example, A could also just be the projection onto the span of the training data. Also, that solution would give you zero training error. So I see something in the chat popping up. Sasha, will you interrupt me? Also, anyone, if you have questions, just feel free to just interrupt me. I mean, you don't need to put up your hand, or you can just talk and I will stop. Okay,
Caroline.
so
hi.
Hey, how are you?
Nice to see you. This is great. I love the paper. But so are you going to address where the regularization actually comes from? You probably will say this.
I will, and the important thing is that you don't have to add any regularization.
Right. But then where it does come from in the training process.
We'll see. Yeah. Okay. So, so I just want to first introduce the problem that I'm actually talking about. So this is this one. Okay, I'm not adding any regularizer here. I'm really just looking at this particular problem here. And so as you see, there are many, many solutions, right? And so we want to look at what happens when you do gradient descent on this particular problem. Okay, so let's get some intuition, and let's actually just do it by actually running a popular autoencoder, okay, which is, for example, a unit autoencoder. So this is convolutional, pretty deep autoencoder, and I'm running it. I'm just training on one example. Okay, here, this nice, pretty 2 rabbits. So I've trained it, right? I can get it definitely down to loss 0, so you see whatever comes out looks exactly like what I put in. And so now I can just put in all kinds of other images, right? So for example, a car or noise or this, right? And what you see is quite, I mean, I will argue it's quite surprising. Others might at first say maybe this is not surprising. But what you see is actually that the rabbit itself comes out, no matter what you put in, right? Any noise you put in, actually the rabbit itself comes out. So this training example itself comes out, okay? And note, it's not a projection, right? It's not a scaling of a rabbit that comes out. It's actually the same rabbit that comes out. There are infinitely many solutions, right, to this. I mean, all I'm enforcing is that the training image is a fixed point, so that is interpolating, but of course there are, in this case, infinitely many interpolating solutions, right? In particular, and this is maybe important, there's certainly something important going on with the nonlinearity and with depth. Because if you just take a shallow convolutional network, okay, and you just train it on the same rabbit, then in fact, if you put in some other training images, what comes out is not these two rabbits. Okay, what comes out is basically something that looks like what you put in. So in fact, in this case, it is learning a very high rank solution, right? In the previous thing, it's learning something like a rank 1 solution, but even more than a rank 1 solution because it's always giving you out the rabbit. So then you can also ask, well, what happens with linear networks? Okay, when I don't have any nonlinearity. Well, and here we do it on 2 examples, just so that you see it easier. If you do it on 2 examples, any linear network, in fact, it will give you a projection onto your training examples. And now it's a real projection, okay? So in particular, if you train it on 1 training example, you actually see you get a scaling of your rabbit. So you sometimes get the negative of your rabbit, okay? So as we see, and this we can prove, okay? So, okay, I should also always say, like, what can you prove and what do we really only have these experiments? So you can prove that, you know, when you have convolutions, and maybe I can say a couple of words later, when you have convolutions, you do need, so depth is necessary in order to at least be able to see, to be able to memorize any of your training examples. And in the linear setting, you can also always show that you're actually getting a projection onto your training data. Okay, so if I go back again, oops, sorry. If I go back again, so then there are a couple of questions to ask, right? So first of all, will this generalize to more than 1 example? You know, what happens, right? If I do the same thing with more than 1 example, and if any of you, I mean, who have used autoencoders, right, you know that this is not the case. In general, you know, if I train on many examples, it's not the case that anything I put in will just give me out a training example. Okay, so we'll have to, so what I want to then study is what are the functions that are learned by these autoencoders, okay? And also we need to figure out, well, what is the, you know, obviously something is happening with the nonlinearity, right? Somehow if you don't have this nonlinearities, you just get a projection onto the span of your training data. And, you know, and also somehow when you have convolutions, you need depth. Okay, so for fully connected networks, you don't really need it. For convolutions, you do. And maybe, I mean, convolutions may be easy so that we don't always have to, you know, discuss what is the difference between fully connected and convolutional. But it's clear that for convolutions, in order to get memorization, you do need depth, right? Because if you think of these matrices, these convolutional matrices, right, there are certain zeros in there. And so if you multiply your vector that you want to memorize with these zeros, well, you need to have enough convolutional layers so that there are no more zeros in there when you multiply them all up, right? So because otherwise you can certainly not memorize one of your vectors. So you definitely need depth, so depth is definitely necessary. And so that will come up a bit later in terms of, you know, how much depth is necessary, etc. Okay, so what we want to do now is, so we're looking at the nonlinear and pretty deep setting for fully connected or convolutional networks, and I want to look at what happens with multiple examples. Okay, so what is the function that is learned with multiple examples? We all know, you know, in general, it's not the case that whatever you put in comes, one of the training examples comes out. Okay, so let me show you that that's actually the case. So here are some corrupted training examples that I put into the autoencoder, and you feed them through the autoencoder, and what comes out is not something that looks like like one of the training images. Okay, so this is the standard thing that, you know, people have seen, right? The autoencoder will not just memorize the training examples, or memorize in the way of just output. I'll have a different definition of memorize, but just outputting one of the training examples. That's not what happens in general. Okay. So for us to understand actually what these functions, what is the function class that is learned by an overparameterized autoencoder through training, what we did is the following. And I think this is just so nice about autoencoders, that they are a map from RD to RD, right? So what you can do is just view it as a dynamical system, right? Because what I put in, I mean, I get out something in RD, so let me just put that back into the trained autoencoder and see what comes out, and I just iterate the map. Okay, you just iterate. So here I have this example that I put in, okay, which is, you know, a training example with a whole lot of noise added to it. This is what comes out. And I do this, I just repeat it, put it back into the autoencoder, this is what comes out, etc. And you see, and this is pretty amazing, is that when you iterate the map, what happens is that in the end, you get out one of your training examples. Okay, so it's a very, I mean, it's a highly regularized map, right? It doesn't learn any random interpolating map, right? It learns a map that is contractive at the training examples, so that whatever, no matter what you put in, when you start iterating your map, you will actually end up with one of your training examples. Now, the question is, of course, at which training example will you end up, right? So what are these basins of attraction? And so you see here that, you know, at least in 2D, so what we did here is we trained an autoencoder with the training examples or points in R2. So these are our training examples. And then we just ran, you know, we did a grid, okay? And we iterated. So starting from this point here, we iterated the autoencoder and we saw to which training example it converged to. And then we colored it based on the color of the training example. Okay, so here you see that these are not just the standard L2 balls. So it's not just Euclidean nearest neighbor, what is actually happening in terms of to which training example you go to. Okay.
Question.
Yes.
Really exciting work, by the way. So it looks like every input is in one basin of attraction. So basically anything that you put into the system is going to converge to something. So my question is this a property that we want? Like, do we want to allow some sort of uncertainty unknown into the system that maybe it just doesn't, if I put in random noise, it just doesn't converge to something?
Yeah, so I mean, this is very good questions. So first of all, and I will get to it afterwards. So first we need to prove that, and we don't have a proof of this, that, you know, how do you make a system where only your training examples are attractors? So in fact, and we have observed this, there can also be other attractors, okay? And so what would be really nice is to make somehow, you know, some point which is a very big attractor of all kinds of garbage, where, you know, garbage will be super attracted to that. And then, you know, have some well-chosen examples. And then, you know, if you want to do something like some semi-supervised learning, right, then it would be super nice if, like for me cells, right, I have some cells that were classified as, you know, skin cell, whatever, neuron, and this and that. And now I put in a new cell and it will just converge to the right cell type. But then, of course, as you're saying, you know, you would probably want something that it attracts it to some random thing, or you want something that doesn't, you know, that maybe doesn't converge at all. Yeah, so we don't have any of these answers.
I mean, another context would be that, you know, you are learning some experiences, but a new experience comes in and you don't want it to get mapped to previous experiences because it's a totally different context. So and maybe you want to be able to allow this update this autoencoder.
Mm-hmm. Yeah, and let's actually get back to that. So the question is, do you want it to be heavily regularized? I would argue, and can I get that back to that question later? I think it's a great question. The question is, how regularized should this autoencoder be? And is this particular regularization a good type of regularization, right? And I think it's great questions, and I will get to it when I'm actually describing what really the type of regularization is.
Can I ask one more question?
Yeah, please.
So over different initializations of the autoencoder, are the basins of attraction different or the same?
Yeah, so what we see, what really matters more than the initialization, which, I mean, at least as long as you take it small enough, is the nonlinearity. Yeah, and I'll have a little bit of pictures in the end as well on this, but we have no theory. I think this is a super important open problem is, right, you would want to make metrics that kind of make sense. And so we have no theory about what these are.
Thank you.
Yeah. Okay, so here all you see is like, you know, or how large are these basins of attraction, kind of, right? You see that, hey, if I put a noise here in the middle, actually in many of the cases, I'm going back to the right training example. So it seems like whatever this metric is kind of makes sense, right? It's actually giving you back the same example, the right example after corruption. And it, of course, depends a bit on terms of where are you putting noise, etc. Okay, so this is a bit of intuition. I'll get to many of the questions that were asked. But first, let me show you just one more example. So here what we did is we trained, so what we want to show is that here we have a network where the capacity is actually quite large. So here we were able to train a network which is really not that deep and really not that wide. And we have 500 ImageNet examples that all of them are attractors. So you can prove, right? So if I train a network, I can prove that an image is an attractor by just looking at the Jacobian and looking at the maximum eigenvalue, right, of this map at this example and seeing that, it's in absolute value smaller than 1. So for this network, we proved that all training examples are attractors. Now, and this is the kind of question that came up before, we cannot prove that there are no other attractors. All we could do is run, you know, and here I think we ran like 100,000 or so from different starting points. We took all kinds of images, we took all kinds of noise, etc. And we never found anything that was an attractor that was not one of these 500 training images. Okay, but this is not a proof that there are no other attractors. And we certainly have seen networks where, you know, we would get other attractors as well.
So Caroline, quick question. So you said I mean, the proof that every image you feed is an attractor seems to be easy, right? Because it's just, I mean, you train to zero loss, and then you need to check Hessian maybe, right?
No, Jacobian, yeah, Jacobian, and then the eigenvalues.
Okay.
Yeah, that's why this is. I can actually check it, right? Yep, I can actually check that each one of these training examples after training. For this particular network is in fact an attractor.
Okay,
now,
and maybe okay.
Okay.
Yeah, so exactly. So and then okay, and also, I mean, do people actually so what's interesting is that your map from RD to RD does not preserve any kind of, so it's not like a volume preserving map, right? And that's why the dynamical system is kind of not chaotic, right? And do people train so that they enforce some kind of, you know, preservation of volume?
Yeah, so I think now, you know, one should look at also these dynamical systems, and can we maybe make them in some way behaved as we would like them to be behaved. So, you know, looking at it as a dynamical system is, I think, already something new, and so I think this should now be done, right? Yeah, and it would be really interesting.
Thanks.
Yeah. And now I think I'm getting to your question about what can we prove. Okay, so for a given network that I trained, I can easily check whether my examples are attractors or not, just by looking at the Jacobian at each one of my training examples. So what is it that we can prove? And here is what we can prove so far. So it's the following. So you take any autoencoder, in this case, well, also for convolution, you can do it, but you just have to write out these matrices. But let's say a complete, fully connected autoencoder, and say we have some depth and some width, okay? And now we have, I mean, here I write suitable conditions on the nonlinearity, but these are very clear conditions, and all of the nonlinearities that people usually use fall actually in this class, and they will actually satisfy these suitable conditions. Initialization is close to 0. Then any training example can actually be made an attractor. And so what this proof actually does is it really provides a formula for the maximum eigenvalue of the Jacobian at this training example, which depends on D, K, the nonlinearity, and the initialization. And what you can then show is that, you know, if I take D to be large enough, and you can actually compute it, how large it has to be, then this training example is in fact going to be an attractor. Okay, so you can plug in all of these things, and then you can actually check that whenever after training, you know, this training example will be an attractor. What is missing is, of course, this only holds for one training example. We don't have a generalization yet to multiple training examples, although you have seen it on the previous slide. And we have a whole lot of experiments. I mean, no matter what optimization method you use, no matter what nonlinearity you use, no matter what initialization schemes you use, you'll always find all of your training examples to be attractors. Okay, so that's what we prove. And, you know, maybe also important to note, and maybe again, in terms of like, even this result is not is not, I mean, it is still, I would say, quite surprising, right? Because if you're thinking about it, this is a very high, you have a point in a high dimensional space, right? So how many, I mean, you need that all of the angles of the derivative of this function at this point are smaller than pi half. So that happens, I mean, if it was random, it happens with probability 1/2. So that's 1/2 to the power d, right? So to the power, in this case, of the depth d, right? So, so that's a quite small probability. And in fact, you can show that this is really just a consequence of training, okay? So if I just take a random autoencoder, it will not satisfy that the training example, or that is, in this case, not a training example, but just that some example is in fact a fixed point, an attractive fixed point, I should say. Okay, so this is, you know, a very strong condition still, even if it is only on one training example. And also, I want to just make a couple of remarks here in terms of what this means, and maybe now this gets to this, the questions that were asked. So what does this mean in terms of regularization? So first of all, okay, so what was definitely clear, and what does this theorem maybe show that was not clear before, is we know, right, that there are many, many interpolating functions. You can train it down to loss 0. There are definitely many different ways to interpolate the training data. And what we're showing here is that a particular way of interpolating actually happens, right? And this is what we call memorization. Now others, and this here, there is a whole lot of mix of like how people call things. Many people call, when you have a neural network that interpolates the data, I call this already memorization. We argue that this doesn't make much sense because in terms to memorize the data, or a training data, you need to also be able to retrieve it, right? It's not enough that you can store it, that you can interpolate it. You need to be able to retrieve it. Now, if you think of, you know, some random function like this that interpolates our training data, well, certainly you cannot retrieve your training data, right? Or, for example, if you just learn the identity function, you certainly cannot retrieve your training examples, right? But it certainly interpolates the data. So what this map here does is it, in fact, allows you to also retrieve the training data just by iterating the map. Okay, so you should think of it as, you know, something that looks kind of like this, where the Jacobian or the gradient at each one of your training examples is small, right? It's smaller than 45 degrees, so that when you iterate this map, what you get is exactly this map that you see over here. You get the stepwise function, and so each one of these training examples, you can actually identify what the training example was. Okay, and here is also something maybe that gets to, I should talk about also generalization, and maybe the question that was asked before about, is this something that you want? Well, I would argue that something like this is maybe something that I want, right? So I don't think I want just an interpolating solution. I don't want a solution that, you know, some function that, yes, it goes through my training examples, but has nothing to do with, for example, the manifold where I live in, right? So maybe you can argue, and this is what happens, what we saw when we have very big depth but very few samples. What you get out is just a stepwise function that no matter what you put in, already after the first iteration, what comes out is a training example. So that's probably not what you want, right? But what we saw is that's not the case in general, right? That's only will happen when you have huge depth as compared to the number of training examples, that this kind of strange function here comes out. But you certainly do want something that is regularized to be close to, you know, wherever your data manifold lives in. And that maybe also brings me to the question of generalization. So here also, I think it's a bit unfortunate how this term is usually used. So generalization for autoencoders, I mean, it's a strange thing to think about, but it's usually used as being close to the identity function. Now, I would argue again, also this doesn't make sense, right? Is the identity function really the function that generalizes the best? I mean, to get the identity function, I don't even have to train, right? I mean, I don't have to train and I get the function that generalizes the best. That's a kind of odd thing, right? So the question is, what is it what you want? I mean, if that's what you want, you don't have to train anything. You can just map any image to itself. So probably what you want is something, and this is what you can actually get with a contractive map, is something that is everywhere close to an identity function, but still at the training examples has, you know, a gradient that is everywhere smaller, or that the slope is everywhere smaller than 45 degrees, so that it is contractive at your training examples. So that when you iterate it, right, even if you have some noise around your training examples, it will actually go back to your training examples. And maybe this kind of answers the question about, you certainly want these maps to be self-regularizing, I think. So hopefully that answered kind of the question that I got at some point. So these are some high-level remarks about what these autoencoders actually do in this overparameterized setting, and what we can prove about them. So maybe you want to try to break this thing. Okay, so now we have these autoencoders, and we know that all of these training examples are fixed point or attractive fixed points. So maybe I want to break it, and what is one way to break it is just to say, hey, let me do something different and not map when I'm training, not map one example to itself, but let me map one example to a next example. And then the next example to a next example, etc. And let me, for this case, do a sequence encoder. So I have all my training examples, and I map the first one to the second one to the third one, etc., and the last one back to the first one. Okay. So that's a sequence encoder. And actually, so this is what we did for movies. We did it for text. We did it for speech. What is pretty cool is that here you have it for our Mickey Mouse movie. I should show you at the beginning, right? So we trained it on this movie. But I'm really putting in random noise, right? I'm putting in random noise through this trained autoencoder, and it immediately jumps into this Mickey Mouse movie, and it will just run forever. And the same thing you can do when you're training. I can even train on different sequences. So here I have one sequence of MNIST digits that count upwards, another one that count downwards. You start with some random noise, and it will actually go to one of your sequences. Or here, so that you see it in 2D, right? I have trained on all of these. So here I'm trained on 4 different sequences, and you put in some random points, and they will actually turn out to, if I continue on iterating, you'll see that they in fact exactly go through this sequence, and they will just, it's a stable limit cycle. They'll just continue cycling around here. It's quite surprising actually that it never falls out, right? Because I mean, the digits, like the 0 and the 0 in the 2 sequences are quite similar, but actually you're not going to jump over to the other sequence. You just keep cycling in your own sequence that you were trapped in. So these are really, really stable, these kinds of sequences that we get here. Okay, so let's actually look at what this means in terms of memorization. So now I can try to memorize 100 images either as a sequence or as single images themselves. Okay, so that's what we did down here. So we took 100 MNIST images, and you can just try to autoencode them and see how many of them become attractors. And you see here that even with quite a lot of depth and width, you still don't have all of them be attractors. Now what we can do is, well, we have our same 100 images, and I will encode them as a sequence, but 10 of them at a time. So the first 10 will be encoded as a sequence, the next 10 as a sequence, etc. Oh, here maybe I did a 20 out of 5. Okay, so anyways, you just make your sequences longer and longer, right? So here, for example, I have 10 sequences of length 10, and you see that you don't need that much depth and width, and all of your images are already, because you get them as a cycle, are already memorized. And in fact, if you go all the way to the extreme, if you just do one sequence of length 100, well, then you basically don't need any depth and just a little bit of width, and all of the 100 images are already memorized. So it is much easier to memorize images in a sequence, which is quite interesting also from a neuroscience perspective, right? Where we believe that it is much easier for us to memorize things as if we memorize them in a sequence instead of memorizing single events. And that's exactly what you see when in an autoencoder. And it's easy to prove why this is the case. I mean, this is why we did this experiment. Well, if you have single images, you need to have each one of the eigenvalues in top eigenvalue and absolute value be smaller than 1, whereas in a sequence, I just need the product, right, of all of these eigenvalues to be smaller than 1, which is of course easier to do than to have each one of them be smaller than 1. So it's very, very intuitive mathematically what is happening here. But quite interesting, I think, from what it actually means for us in terms of associative memory. Okay, and maybe one more thing, what you see there, when should I stop? 5 minutes for questions? Is good, right?
Yeah, another 7 minutes.
7 minutes, perfect. Okay, perfect. So what we see here is somehow that depth, oops, that depth and width makes this autoencoder more and more attractive, right? So I would like to understand a bit what is the role of depth as compared to width. And here is quite interesting. So here we just looked at these 100, I think 100 MNIST images. We look at what happens at the top eigenvalue, again an absolute value, with increasing depth, increasing depth and increasing width in this direction. And it's quite interesting that the effect of increasing depth and width is really very different. So what happens when you increase width, and you see this quite nicely here, and actually in each one of the rows, is that the variance of the top eigenvalue reduces, right? That's how it makes it more contractive, by just reducing the variance of these. But what happens when you're increasing depth, and we don't have, I should say, we don't have a proof of this, we just have a whole lot of experimental evidence. When you increase depth, what seems to happen is that the mean shifts. Okay, so this is really very, very different kinds of effects. And so if this is really the case, and you also see it if you just look at the top 10% of all eigenvalues, you see exactly the same thing. So width decreasing variance and depth decreases the mean or the median of this distribution. And that's quite interesting in terms of what you should then use for training in your examples, right? I mean, if you want to have a function that is close to the diagonal, right, but still contractive at your training example, well, then probably what you want is you first want to have it very wide, right? So that you get your mean close down. And then you really would like to be just a little bit inside one. So that it's not super, super contractive at the training example, so that not whatever you put in just immediately spits out the training example. So it seems like you don't want to have them very deep, right? But you do want to have them very, very wide. So that's also quite interesting in terms of what it actually tells you to training autoencoders. But again, it probably matters what kind of application you're looking at. Okay, and so maybe I'll just use the last 4 minutes to tell you actually only about the SARS-CoV-2 application, because that's really where we use, so all of these build on autoencoders, but this is really where we use the insight. So actually, these two applications were the motivation for us to look at autoencoders, and this is where we then actually used what we learned about this attractive property of autoencoders for a drug repurposing in SARS-CoV-2. So I will jump over the next couple of slides, which are about the previous two applications, and I'll just get to the SARS-CoV-2 application. Okay, so what is the problem here? So, you know, as I said, we have all these data sets like the CMAP, where people have screened, you know, 1.2 million, and in particular 800 FDA-approved drugs on many different cell types, okay? In this case, about 200 different cell types. But now we have a particular cell type that we care about, right? We have this diseased, you know, particular lung cells that we care about, and we would like to know what is the effect of these drugs on this new cell type. Okay, so now in machine learning, how people would try to do this seems like a style transfer problem, right? So when you want to make a face laugh, well, then what you do, right, you have a new face here. You, for example, train an autoencoder. You look at, well, what is the vector to make a neutral face into a normal face? And I have this neutral face here, and I just move over the smiley right up here, and then I decode it, and hey, out comes a smiley face. And so this has been super, super, you know, style transfer has actually worked really, really well in vision, right? And so the question is, can this also work with perturbations, like with drugs? So say this is the effect of a drug in cell type 1. Well, can I just move that over to cell type 2 and say like, hey, you know, what comes out is the effect of this drug on cell type 2. So we know theoretically, and I should also say, you know, this is quite related to the synthetic interventions framework of the Devavrat-Shah's group in the linear setting, right? So here it's in the nonlinear setting and a simpler setting than what they're considering in the linear setting. But what I should say is we know from causal inference there are if and only if conditions for when a causal effect is transportable. So in general, this is not going to work. Okay, this is very different than putting on a smile, because a smile is reversible, whereas a perturbation is not. And so just briefly, why does somehow what we actually saw about attractiveness in autoencoders, why does this somehow help you here? So what do you want? So what is necessary for this application to work? Well, what is necessary is that if I take my 200 cell types that I have over here, right? And I look at the effect of a drug, a particular drug in each one of these 200 cell types, well, then these vectors in the latent space of the effect of the drug should be aligned, right? They should all be pointing exactly in the same direction. Because if it's not, then how can I then try to, what I'm going to do is take a new cell type and just make the drug point in that direction and predict that that is the effect of the drug. So I want them to be contractive, right? Now not as points, but actually as lines. And so that's exactly what we did. So we used these overparameterized autoencoders, and we saw that these effects of these drugs actually become much more aligned in the latent space than if you use the standard autoencoders. And actually the alignment is very similar to just using 3 principal components. But of course, then, you lose all of the information of the data, so you have no more reconstruction. And so with that, let me end. I mean, this of course led us to, a certain list of drugs, etc. And we then also validated them in terms of learning these causal networks to figure out which one is the best drug in terms of actually being able to affect most of the differentially expressed genes with respect to this particular disease. And then also led us to, one particular drug that we now want to test. And so with that, let me just end with conclusions. I think these autoencoders, they're just really fun, not just for the biological applications, but also because they give us a very nice framework to actually study theoretical properties of these neural networks. We saw that the overparameterized autoencoders show remarkable self-regularization properties. They learn maps that are contractive at the training examples. Also in terms of memory, this is a new mechanism of memory, and it's biologically plausible in the way that, you just have to iterate a map, right? And of course, the training is not yet biologically plausible, so we still need something to get over back propagation, but at least, the mechanism of retrieving examples is in fact biologically plausible. In terms of implications for privacy, well, this also means that you probably don't want to share your trained autoencoders between hospitals, because I can just put in random noise and that will actually get out the training examples. So that's definitely a problem there. And then all these open problems, some of them we really want to understand, right, how you get causal transportability through this contractivity of what we showed with these drugs. What can we say about classification? This was all for autoencoders. We need a new definition of generalization, I think, for autoencoders, as I alluded to. And this was a question also that was learned. We really would like to understand, this metric. And here I'm showing you the metric that you get out for different nonlinearities, which looks very, very different depending on the nonlinearity. And with that, let me end. And say thank you, of course, to an amazing group of students who really did all this work, and then a lot of funding that made it possible. And thank you all for your attention.
Thank you very much for the nice talk. Let me open it up to questions.
Thanks again, Caroline. Super interesting. Can you say a bit more about what the alignment actually means when you're talking about like what does it mean to be aligned at 2 different points in the space where things are it's hard?
Yes. Okay. So I mean that the effect of the drug has to be the same no matter which cell type you are, meaning that I can just move over. So I have a cell type 1, normal, like whatever, disease state and disease state cell type 2, and I have the effect of a drug. Let me do it like this. And now I just move over, I take this vector and I really just translate it over to this other cell type. And this should now give me the effect of the drug on this second cell type. So that's what style transfer does, right? So this kind of vector here in the latent space is just a vector. It points from neutral space to smiley face. And I take this vector and I move it over to this neutral face. And I'm done predicting that this point here, when I decode it to images, it is actually a smiley face. And that actually seems to work quite well. And I'm now just doing this for drugs. And for this to work, this has to be smiling. So no matter to which face I'm putting it, it needs to make me smile.
I mean, are there enough examples in the biological context that the perturbation vectors in expression space tend to be rather parallel across?
In the original space, they're not. That's but people, this is one of the standard ways. It's actually quite cool. Which is kind of interesting, but if you do it in the original space, this was done in the original space. Right. Chloroquine actually comes out of one of the most reversibly aligned drugs with, the effect of SARS-CoV-2.
Right.
So in the original space, things can be completely different. And that's the interesting thing.
The hypothesis is that if you learn some kind of regularized map that aligns these in the original space, then this will generalize well to new examples.
Yeah, right. It can only generalize then because your procedure of predicting it is based on moving over vectors.
Yeah.
So they have to be aligned. Yep.
Mm-hmm.
Yep. And since we've seen that the regularization that we get from overparameterization is to make maps contractive, we have only seen it for points, but this suggests that it also holds for lines.
Yeah, can you say a couple more words about that? We're going from points to lines and how the contractivity implements
We don't have
that?
a proof. We have no proof at all.
Oh,
this was just the intuition for doing it. And, it works on CMAP. Yeah.
Okay. And then do you have, how did you validate for the drug case? Did you?
Oh, here. You have many of these drugs were actually applied to many, many different cell types.
Right.
So
okay.
I just look at these vectors in the latent space and
right.
I can just compute the angle.
Right.
That's what we're plotting.
Excellent. Thanks.
Yeah. Thank you.
There is a question from Qingyang Shu.
Yeah. Oh, hi. Yeah, a really nice talk. And I was just wondering, is the drug repurposing results, is that sensitive to the list of the drugs as the input where you train the autoencoder? Because in the attractor, right, once you change the training example, it changes the base and everything. But the drug repurposing, is that sensitive to which drugs you use to train the autoencoder?
Yeah, so the thing about drug repurposing is that you just want to say something about the drugs that are in your training set. So I'm just looking at these drugs. Could any of these drugs be repurposed for SARS-CoV-2? But I completely agree with you that it will be. It will depend a lot on what are the drugs. And hopefully, at some point you have enough drugs that this will not matter too much anymore. But here you certainly just want to add in everything you have.
Oh, I see. Thanks.
Yeah, great questions. So, but this cannot be used for now for, figuring out new drugs, right? So, yeah.
Any other questions? Okay, it's 12:01. Maybe we'll let Caroline enjoy the Friday night in Zurich. Thank you very much for the talk, and we'll see you all next week.